Good day, Honors Biology students. Welcome to video lecture episode 24. Today we're continuing in section 3 of chapter 12. Remember, last time we were together, we talked about the central dogma, and in particular, we talked about transcription. Well, now we're going to talk about RNA processing. This starts on slide 25. When scientists compared the coding region of the DNA with the messenger RNA, they made a discovery. They noticed that the messenger RNA code was significantly shorter than the DNA. What they discovered upon further examination is DNA is interrupted periodically by sequences not in the final messenger RNA. These little sequences, intervening sequences, are called introns. Introns are non-coding regions and are therefore removed, hence the reason they are not seen in the final messenger RNA. In the sequence, the ones that are left are called the exons, which are, of course, the coding regions. All right? Now, besides the sorting of keeping the exons and removing the introns, there is two more RNA processes that go on. One is adding of a protective cap on the 5' end, and the other is the adding of many adenine nucleotides, which collectively are called a poly-A tail, which are added to the 3' end. So these other two processes go as follows. There's a protective cap put on the 5' end, and there's on the 3' end, there's a poly-A tail, which is made up of many adenine nucleotides. Now let's talk a little bit about the code, a little bit more of the DNA code that is. Scientists recognized the only way the DNA varied among organisms was in the sequence of the bases. This was recognized as there was at least 20 amino acids used to make proteins. Now let's connect this to math. Scientists first hypothesized that one base coded for one amino acid. Following this logic and using math, they deduced that 4 bases could only code for 16 amino acids. Then they thought if a group of 3 bases coded for one amino acid, then there would be more than 20 codes, that being 64, in other words, 4 cubed, in fact. Well, in the 1960s, via experiments, scientists concluded this is the case, and that 3 bases in combination did form an amino acid. These triplets, as they are referred to as codons. And if you look at figure 12.14, which I have a similar image on slide 31, it lists all the combinations for the 20 amino acids. Okay, this, you can title this if you'd like in your notes. You can call it the Dictionary of the Genetic Code. All right. Now, while illustrating that, did you notice that only 3 codons are specific for an amino acid? These are the stop codons. They are UAA, UAG, and UGA. Now, in addition, there is only one codon, which is called the codon which starts all proteins and enzymes, and that is being AUG, which is methionine. If you look at the image both on slide 31 and 32, you can see what I'm referring to as well. So AUG is the start codon, and there are 3 stop codons, UAA, UAG, and UGA. All right, now let's move a little bit more into the code, and let's talk about translation. Now we move to the second stage of the central dogma, that being translation. Remember, transcription occurred in the nucleus and went from DNA to messenger RNA. Once the messenger RNA leaves the nucleus via the nucleopores, it then heads into the cytoplasm. After it is in the cytoplasm, the 5' end of the messenger RNA connects to the ribosome. After the messenger RNA is attached to the ribosome, transfer RNAs come in with the anticodons that complement the codons on the messenger RNA. The code in DNA and RNA is read 5' to 3'. The anticodon is 3' to 5'. They begin with AUG, ending in one of 3 codons, UAA, UAG, or UGA. Now, if you look on slide 36, you can see more graphic representations of what we've been discussing, all right? So now let's talk about the role of the ribosome in relation to the code. Well, we're going to briefly mention that role, just real briefly, okay? The ribosome itself is actually made up of 2 subunits. You can see this on slide 37 in the image that I've provided there. When the messenger RNA has left the nucleus, the 2 subunits form together, attaching themselves to the messenger RNA. Once the ribosomal subunits are attached to the messenger RNA, the transfer RNA with the anticodon CAU can come carrying methionine. And remember, this is that start at the beginning, right? Well, this begins the process of translation. From here, transfer RNAs will bring anticodons until one of the 3 codons is placed. Once finished with translation, the enzyme protein is then transported where it is needed in the body. Now let's talk about something that I think is really important I'd like you to know in connection, and it's called one gene, one enzyme. At this point, scientists had learned that DNA works as a code. Now the next step is to understand the relationship between the genes and the proteins they code for. This brings us to the work of 2 scientists, George Beadle and Edward Tatum. And you've probably heard those terms before if you've done any searching about DNA. In fact, you'll hear it usually referred to as Beadle and Tatum's work. Well, in 1940, these scientists worked to provide evidence that genes code for an enzyme. To do this, they studied mold spores which were mutated via exposing them to X-rays. The mold spores studied were Neurospora. These particular mold spores can normally grow on an artificial medium. In other words, a medium is an area where they can grow and get nutrients from. While this artificial medium provides no amino acids, it is called minimal medium. In the experiment, Beadle and Tatum exposed the spores to X-rays and grew them on complete medium. Complete medium has amino acids, unlike minimal medium, and thereby provide all the amino acids Neurospora need. If you look on slides 43, 44, and 45, I just have some fun little images of X-rays. I thought it'd be kind of cool to show you. All right, so let's just continue our discussion. We're just about there and finished for the day. Well, they, referring to Beadle and Tatum, compared this by taking the mutated spore and growing it in the minimal medium. The mutated spores were unable to grow in the minimal medium. They proceeded to determine what amino acids was missing. To do this, they grew the spores on a minimal medium with a supplement such as arginine, and I do have a picture of what arginine looks like on slide 46. Beadle and Tatum hypothesized the mutant spores were missing an enzyme needed to synthesize arginine. From this, they developed the one gene, one enzyme hypothesis. This hypothesis has been modified due to our understanding that polypeptides make up enzymes, and this modification is one gene codes for one polypeptide. All right, ladies and gentlemen, this concludes our lecture today. If you have any questions, you can leave them in the comments below. Send me an email. I hope it's been helpful. Have a nice day, students. Take care. Goodbye.